Hello everyone, this is Phanidhar Kukutla from the laboratory of Dr. Jiannong, Department of Biology at New Mexico State University. We are working on the metagenomics of mosquito gut ecosystem. Today, we are going to demonstrate to you a process to deplete ribosomal RNA from the total RNA samples to enrich messenger RNA for RNA-seq. RNA-seq is a widely used technique to study the functions of microbial community. Firstly, I'm going to demonstrate to you how to isolate total RNA and metagenomic DNA from mosquito gut.
Anopheles gambia mosquitoes are maintained at an insectary. Mosquitoes are first fed on sugar meal before, and then afterward female mosquitoes are fed on mice for egg production. These mosquitoes are fed on sugar, and these are fed with mouse blood. Prior to the procedure, all dissection tools and slides were sterilized. The mosquitoes are surface cleaned with 70% ethanol 3 times in a petri dish and placed back on a glass slide to dry out the ethanol. Place the mosquitoes on a different glass slide and put a drop of sterile 1x PBS solution. Then, gently pull out the gut. The mosquito guts will be used to get metagenomic DNA and total RNA, respectively. Before we begin the RNA work, it is important to take standard precautions to avoid RNA degradation by maintaining a ribonuclease-free environment. While wearing gloves, clean the work area, pipettes, and all required apparatuses with RNA-seq.